Okay. Hello, everyone. I'd like to thank the organizers for inviting me. What I'm going to talk about today is some work I'm doing in my PhD, studying gastrulation in the mouse. So during early development, there are two main phenomena that take place. One is a massive increase in cell number, and the other is an increase in the diversity of different cell types that we observe. At around day 6.5 days after fertilization of the zygote, of the oocyte, whatever, we have an embryonic structure which looks something like this cup shape here. And there are three tissues. Two of these are extraembryonic and will make supporting tissue. And the remaining tissue is called the epiblast. And this is what's going to give rise to all the cells that go on to make up the adult mouse. The first few decisions that the epiblast cells take on the way to doing this is called gastrulation. And it's such a critical point in development that a famous embryologist, Louis Wolpert, has remarked that it's not birth, marriage, or death, but instead gastrulation, which is the most important time of your life. And he's not wrong, I suppose. Okay. Along with gastrulation, at the molecular level, which you can guess I'm probably going to talk about next, there are a large number of anatomical changes that take place. What happens first is that these epiblast cells, which lie in the middle of the embryo, here we're looking at it from above, as if we'd sliced through, a number of them migrate towards the posterior side of the embryo and form this ridge-like structure called the primitive streak. Some of these epiblast cells then ingress through the primitive streak, moving from an epithelial state to a mesenchymal state. These new cells are called the mesendoderm, and they're illustrated in the red here. These cells are going to go on to give rise to two of the germ layers, the mesoderm and the endoderm. If we now change to look through the side of the embryo rather than the top, we can see these mesendoderm cells flank out to the wings of the embryo. Then the endoderm cells will push to the outside of the embryo, and then we'll see a large number of much more complex morphological changes that builds up the mammalian body structure as we know it. So this is what happens anatomically, but we already kind of know what happens anatomically because we've seen it down the microscope, for example. What we don't know is what's happening molecularly to all of these cells through development. And so this is what we're trying to assay with this project. We've harvested embryos from each of these time points, so from 6.5 days after fertilization to 8.5 days after fertilization. At day 6.5, we've got only these three main cell types I mentioned at the beginning. Meanwhile, at day 8.5, we have very many cell types that are present, including the beginnings of the organs. We've taken embryos every 6 hours during this time course. This is a very continuous sampling, and of course it necessitates experimental scientists getting up at midnight and 3 a.m. to do these experiments, so a very hard-fought battle to get this done. But we have a very continuous reconstruction, effectively, of development. We've collected these time points into various Tyler stages. These are anatomically rather than temporally defined stages of development, and they match up to these images that we see here. The final data set is composed of over 350 whole embryos. This has taken a lot of time to gather. And I'd also like to point out that it's very challenging to actually collect these embryos in the first place. Here are some embryos from our first time point, day 6.5. That scale bar is only 1/4 of 1 millimeter long, and these need to be dissected out of the uterus of the parent mouse. Okay, so now we have these embryos. We have to think about how we're going to study them. Cellular decision-making, which is effectively what is happening during development, is inherently a unicellular process. Single cells look at the signals they're exposed to, they look at their own internal state, they integrate this information, and then they make some decision. If we're going to study this, we need to do it at a single cell level. And so for this reason, we've applied single-cell RNA sequencing to the cells from these embryos. We dissociate them into a single-cell suspension, and then put them into this 10x chromium machine, it's called. This captures single cells in tiny nanoliter droplets, along with reagents and oligonucleotides, so that we can have massively parallel library preparation for single cells separately at the scale that actually matches the number of cells that are present in these embryos. So by the time we've done QC and all of that jazz, we're left with about 94,000 cell transcriptomes. These are quite high quality for droplet data sets. We have on average 15,000 unique RNA molecules that we've captured for each cell. And these 15,000 molecules translate to generally around 3,500 unique genes per cell. And here are those 94,000 cells. They're projected with a method called t-SNE. What this means is that every point here is a cell, and every cell is positioned close to other cells that share a similar transcriptional profile. The cells are colored by the top-level cluster to which we've allocated each cell. And so in terms of the developmental story, we start here with the epiblast cells. That was that tissue that I said was present at day 6.5. The cells that ingress through the streak are found up here in this tan cluster. Those are the mesendoderm. And then emerging from the mesendoderm, we see on the one hand a big group of endoderm cells, where at the end we have the beginnings of the embryonic gut. Meanwhile, in a different direction, we have early mesodermal progenitors. The mesoderm will go on to form a lot of structures in your body, including, for example, your blood and your muscle. From the mesoderm, we can see a few very obvious trajectories coming off of this plot. First, there's all of this orange and red group of cells here. This is the hematoendothelial lineage. This is the embryonic blood, and also the lining of the blood vessels in the embryo. Over here, we have the allantois and also other extra-embryonic tissue. This is going to go on to become the umbilical cord. And over in these blue clusters here, we have a set of other mesodermal tissues, very heterogeneous, but it includes, for example, somites, which make up the ribs and the backbone of the animal. Not all of those epiblast cells actually go through the streak. Some of them remain epithelial, and they age and they become the ectoderm, which will become your brain and your skin. And this is represented by these green clusters over here. Finally, in the embryo, we have these cells over here in yellow. These are the neuromesodermal progenitors. Usually, you might expect cells to commit to one of the germ layers and then remain within other cells of that germ layer, right? They've committed to that fate. They can't jump between them. But these are particularly interesting because these cells can give rise both to cells of a mesodermal character and to those of a neuronal character. So it's appropriate then that we see them joining up effectively the mesodermal side and the ectodermal side of our plot. Because we've captured whole embryos, we also have extra-embryonic tissue, which is present in these two groups of cells here. And then there are a number of other embryonic clusters which have very distinct transcriptomes, for example, cardiac muscle. I've just told you this story about how all of these cells arise over time, but you don't actually just have to take my word for it, because we know the time points that these cells were harvested from. And this is what we're showing down here. So this is the same T-SNE, and now the cell is colored red if it originated from the time point indicated in the title. And what we observe initially is that the cells occupy a relatively small area of the plot, and you'll also notice that it's quite homogeneous in structure. By the end, the cells have spread out to all the extremities, and you'll notice there's a lot of fine structure in the plot. This represents a higher level of molecular heterogeneity, as we would expect from the developed embryo, and this, of course, matches up to the increased anatomical and morphological complexity that we observe in the embryo through this time course as well. So what else can we do with this real-time information? We can ask, at each time point, what is the frequency at which different cell types exist? And this is what we've plotted here in this ribbon plot. So at the start, for example, we have lots of this brown epiblast cluster, which has almost entirely disappeared by day 7 3/4. You can also notice that at the beginning, we have very few cell types, as we expect, and then by the end, we have a very diverse set of cell types. You can also notice at a high level that there are some cell populations which are only transient. These early mesodermal progenitors have all gone by day 8, as have the neuromesodermal progenitors by our final time point. Now, the colors I've been showing you so far, these have been the top-level clusters that we assigned. However, each of these clusters actually contains a lot more structure within them. And to try and uncover this structure, we've performed a second round of clustering within each of these clusters. We've then manually curated these subclusters into a list of the different cell types that exist at the different time points in our data. And this legend up here is each of those. And I just want to point out to you that there's a lot of different cell types present in the embryo. And this makes visualization particularly challenging. The sort of plots I showed you before don't work so well when I have 40 colors, which are all just slightly different from each other. So instead of visualizing this at the per cell level, it can actually be more effective to visualize it at the per cluster level. And to do this, we've implemented a method from Fabian Theis' lab called graph abstraction. This relates clusters to each other by the overlap of the cells of these clusters in the gene expression space. So clusters, here each point is a cell type here, or a cluster. They have a dashed line between them if they're close to each other effectively in gene expression space. And again, we can reconstruct the same developmental trajectories that exist within the data. If we start from the epiblast here, we have on the one hand all of the ectodermal tissues. If we move towards the mesendoderm here, we have all the endodermal tissues down here. And finally, all of the mesoderm sits over here. And once again, by referring to the real time, we can confirm that what we're saying is correct. All of the most developed cell types are present around the edges of the plot, and all of the most basal or youngest are in the center. So what can we do with this kind of data? On the one hand, we can just look at the gene expression information that's encoded within the data set. So here, for example, I've taken a subset of some mesodermal progenitor cells, the hematoendothelial progenitors, and then the blood and the endothelium. The endothelium is going to be the lining of the blood vessels. And the cells from this cluster, these clusters, are laid out here in a diffusion map. So we can see in the top left, the mesodermal cells give rise to this spout of blood, and also to these endothelial cells shown in orange. We can take, for example, these blood cells and make an ordering of them that matches up to how developed these cells are. So we order them, on the one hand, from the youngest, transcriptionally the youngest looking cells, and transcriptionally the oldest looking cells, and then a continuum between them. Then we can look at how different genes are expressed along this ordering. For example, we see hemoglobins, what you expect to see in blood, very highly expressed at the end, but also more subtle features, like this transcription factor RUNX1 is expressed only transiently. Now we have this this is quite a simple example, admittedly, this is just the blood. But we have this kind of information for all of the trajectories, so not just the blood, not just the endothelium, but everything that's happening in the embryo between these time points. So there's a lot of understanding we can get of molecularly how cell fates are specified, and perhaps how you could go in the other direction, from a regenerative medicine kind of sense. So that's one way to analyze it, was to look at the data just by itself. But you can also use this as a reference for other experiments. TAL1 is a very important transcription factor in specifying the embryonic blood. The problem is, if you knock out TAL1 from an embryo to see what happens when it goes wrong, the embryo can't make blood, and so it dies. So you don't get to look at these TAL1 cells after they've missed their blood commitment, because they're already in a sick embryo or an embryo that's dead. So to get around this, we've made what are called chimeric embryos. We make a stem cell line, a mouse embryonic stem cell line, where the gene is knocked out, and the cell also constitutively expresses a fluorescent protein. We use TD tomato. Cells from these stem cell lines are then injected into very young embryos called blastocysts. The blastocysts are returned to a parent mouse, where they're then allowed to grow to an older age, to time points, in fact, within our time course. So we allowed E7.5 and E8.5. And now, even though the tomato positive, the knockout cells are present and can't contribute to blood, the embryo won't die, because blood can still derive from the wild-type cells. What this means is we can then capture cells from these embryos, we flow sort them, so we now know the background of each cell transcriptome, whether it was from a knockout background or a wild-type background, sequence them and analyze them. So now we can look at what happened to those cells which were missing TAL1 without the embryo dying. So just some high-level features here. We have cells from these chimeric embryos. Over here, they have TAL1, and over here, they're missing TAL1. And first of all, we notice that there are no extra embryonic tissues present in the TAL1 knockout cells, from the knockout cells. This is because they are mouse embryonic stem cells. They are primed, which means they can only contribute to epiblastic fates. And then at the later time point, we see there's no blood in the TAL1 knockout, as we expect, because that was the whole point of knocking out TAL1. Now we focused in on one specific lineage. We focused on the hematoendothelial progenitors. These are the cells that are going to give rise to the blood, which is what TAL1 is knocked out, or alternatively, the endothelium. If we look at this plot is the t-SNE that I showed earlier, the atlas. A cell is highlighted if it is the closest match in the atlas for one of the cells in our chimera. So these cells that are shaded in black are the closest matches in the atlas for the cells that derived from the chimeric embryos from the cells that were wild-type background. The red ones are the same, except they're the matches for cells from the chimera that were TAL1 knockout background. First of all, you'll notice that there are cells from the wild-type background in the chimera that are committing toward the blood lineage, as we would expect. Without TAL1, there are none. So that's good. But also notice that these TAL1 cells are not just alternatively committing towards the endothelium. They are visibly more basal in this t-SNE. This marries up with other information. So if we look at the stages of the cells to which these TAL1 knockout background mapped to in the atlas, they are almost all of a younger stage than the ones that the wild-type background cells mapped to. And also, these TAL1 knockout cells co-express early mesodermal markers. So there's a whole bunch of evidence here that's pointing towards the fact that this knockout doesn't just stop them becoming blood, but it's actually slowed down their development entirely. And so that's an example of how we can use it to map or classify other cells to this data. You might also imagine, perhaps you have an in vitro system where there's an organoid, perhaps, or a gastruloid, as some people call them. You can actually compare your cells to our reference here and see how your system compares. Is it a good match? Is it a poor match? How is it similar? How is it different? And so on. We can go in. We can look at those developmental trajectories, like I showed very briefly, with the blood. And finally, if perhaps you're a developmental biologist who has a particular gene or gene regulatory network or cell type that they're particularly interested in. You can take our atlas, you can subset cells out of it, and you know that these cells are behaving in the proper way because they were from their real in vivo environment. They were exposed to the proper set of signals that you would expect in a real embryo. So I'd like to thank the Wellcome Trust, who funded my PhD and also funded this project. And they fund it through this big group of PIs from Cambridge in the UK. And specifically, John and Jenny and Bertie have been working on this project. On the analytical side, these 4 have been helping me out, while on the experimental side, these 3 have been leading the project. And I'd also like to thank the rest of the Marioni group, who make work quite fun. Thank you very much.
That's a really nice talk and wonderful work. What computational method are you using to label the cells? I'm really curious how you got all those wonderful labels on your cells.
So you mean the clustering, for example, in the TSNE?
No, not the TSNE, but the actual labels on the cells, the different cell types, right? You have different colors for different cell types?
Yes, so those were just top, you mean, so these, where we had like 40 different colors, or where we had about 9 different colors?
Yeah, all the colors on the right-hand side that you assigned to the cells. How
so
are you assigning those to the cells?
We just manually selected them, I'm afraid to say.
How do you manually assign them?
Oh, I see. So do you mean, how are we labeling the actual cells? We build a graph where we connect cells by their proximity in gene expression space, and then we use graphical clustering tools. There's one called Louvain clustering, which is quite effective. It sequentially merges groups of cells together, maximizing this score called modularity. Modularity reflects what share of connections are retained within clusters compared to going between clusters. And so you just try to optimize groupings of cells like this.
And then you look at the clusters and then you manually label them?
Yeah, looking at marker genes and we go, yeah, this marker gene is expressed in this cluster, therefore it is endoderm.
Thank you.
Could I ask a follow-up question to here?
Yes.
On your right. So the boundaries between these clusters seem to be quite continuous. In other words, the boundary between brown and light brown, for example. I'm curious if your algorithm actually decided on those boundaries automatically. You basically told it, find me 40 clusters, and it found those clusters. Or did you have to manually adjust where one cell type ends and the other one begins?
So you don't actually specify the number of clusters. But ultimately, I just wouldn't bother paying too much attention to exactly where the line is drawn, because these cells exist in a continuum. Because we've sampled so regularly, the cells from each time point overlap with each other effectively in their amount of development, and therefore you could draw the line here, or you could draw it here, or here, or here, and ultimately, as long as you're capturing the broad patterns with these clusters, I think that's...
And in that continuum, you find that the cell types that emerge are fairly meaningful. It's not like you could say, well, maybe there's twice as many clusters and so on and so forth.
So, I mean, so these are very broad clusters. Like you can see, for example, some of this stuff is called late mixed mesoderm, because it actually contains many different cell types. So these are quite broad, but we're aware that they're broad, and that was the point of the second layer of clustering.
Well, we're getting very technical about your clustering. Did you use Jacquard before Louvain to enhance the modularity?
Probably not. I don't know. To be honest, I haven't heard of this version of the clustering algorithm, I'm afraid. You should. Okay.
Right here.
So there's a lot of polarity developed in the embryo by gastrulation. So, for example, in what you classified as the streak, can you tease apart the anterior-posterior polarity of streak cells?
We should be able to do this. Previous work where, an old postdoc in a group called Antonio, he processed the data set where they sorted out specifically epiblastic and mesodermal cells and then sequenced them in plates, so lower throughput. And that's exactly something they did in that paper. So, I mean, in principle, we should be able to do it here, but we haven't done it now.
In the TAL1 knockout, where the blood is missing, I think you said that the extraembryonic tissues are also missing. Is that right?
So they're present, but only from the wild-type background cells, because the injected embryonic stem cells cannot contribute.
Okay. So does it mean that the extraembryonic tissue that originates the blood lineage, in part?
I suppose that would be an interpretation from what I've said, but I don't think because we know
it's actually controversial in the literature.
Oh, okay.
Yes.
So I think
so if you have a gene that knocks out the blood, and at the same time extraembryonic tissue are missing, which are amongst the earliest tissues generated, doesn't follow that those tissues generate the blood?
That's a good point. But But it's probably also not, it's not the case that all blood is derived from extraembryonic tissue, I would imagine. And therefore, we can still study the cells that are in this confused slowdown state.
Well, it's a wonderful opportunity to study these questions.
That's a good point. I will definitely remember this. Thank you very much.
Brilliant. So I'd like to thank all the speakers in that session. It was a really great session. So thank you very much.